Hello students, welcome to EPG Pathshala. I am Ramesh Kothari, Professor of Microbiology, UGC CS Department of Biosciences, Saurashtra University, Rajkot, Gujarat. Welcome you all. Today we are going to discuss about regulation of citric acid cycle from the paper Metabolism of Carbohydrate. We know that cell is a living chemical factory. At a time in a cell, 1,000 reactions are going on. Some are anabolic, some are catabolic, and some are amphibolic. Somewhere in cell, some macromolecules are undergoing catalysis to generate several monomers, which in turn can be used to build up other macromolecules. Many times this macromolecules is used for synthesis of other macromolecules. So, for the synthesis of various macromolecules from monomers and monomers from macromolecules, a regulation of all metabolic reactions is an important task for a cell. Almost all carbon that accumulate in the carbohydrate degradation is converted by pyruvate dehydrogenase complex to acetyl coenzyme A. The acetyl coenzyme A is also formed by different enzymes in the degradation of fatty acids and of ketogenic amino acids, and it therefore is a central hub in energy metabolism. The next step towards complete oxidation in the citric acid cycle, also referred to as Krebs cycle or the tricarboxylic acid cycle, the basic idea of TCS cycle consists in releasing the substrate carbon as carbon dioxide while retaining the substrate hydrogen for cold combustion in the respiratory chain. In the complete degradation of pyruvate dehydrogenase, which is also known as PDH popularly, and the citric acid cycle perform the oxidation of all substrate carbon to carbon dioxide. The hydrogen is retained in reduced form. It is subsequently oxidized in the respiratory chain. Looking to the objective of this lecture, we will discuss how citric acid cycle is regulated by different mechanisms. We will also discuss about the different levels of regulation and process of regulations and its importance. Further, we will also explore the underlying biochemistry of citric acid cycle regulation. As we know that citric cycle have various reversible and irreversible reactions. Mostly reactions are reversible, and at different level energy is generated in form of ATP, and how a cell is able to produce this amount of energy through this several reactions and how all reactions are regulated, that we'll see in detail. This figure shows the concept map of citric acid cycle. It is a chain of chemical reactions used by aerobic organism for the production of energy through the oxidation of acetate derived from carbohydrates, fats, and proteins, and converted into carbon dioxide and chemical energy in the form of adenosine triphosphate. Citric acid cycle has importance to many biochemical pathways, indicates that it was one of the earliest component of cellular metabolism and may have originated abiogenically. In eukaryotic cell, the citric acid cycle occur in the matrix of the mitochondrion. In prokaryotic cell, such as bacteria, which lack mitochondria, the citric acid cycle reaction sequence is performed in the cytosol with the proton gradient for ATP production being across the cell surface, that is plasma membrane, rather than the inner membrane of the mitochondria. If you see in the figure, the step 1, that is acetyl-CoA production, which is take place in the cytosol, and it is then transported to the mitochondrial matrix during stage 2, where acetyl coenzyme A undergo oxidation and enters in the TCA cycle. Stage 3 of electron transfer and oxidative phosphorylation also take place in the mitochondrial matrix, where the electron transfer chain results in the production of ATP at the stage of ATP generation by electron transfer chain. Thus, the amino acids, fatty acids, and glucose find its way through acetyl-CoA, acetyl coenzyme A, to enter in the citric acid cycle, which results in the formation of generation of ATP, carbon dioxide, and reducing power. Sir Hans Adolf Krebs, a German-born British physician and biochemist, was the pioneer scientist in study of cellular respiration, a biochemical pathway in cell for production of energy. If you see in the figure, Hans Krebs gave the reactions of TCA cycle. Looking at the reaction of TCA cycle, very first reaction is condensation of oxaloacetate with acetyl coenzyme with the help of acetate synthase enzymes. It produces citrate. Here water molecules is added, coenzyme ASH is released. Now the citrate converted into aconitase by the enzyme. Its reaction is dehydration reaction, and enzyme is aconitase. Now this aconitase is converted into isocitrate by the enzyme. This reaction is again hydration reaction. This isocitrate is again converted into alpha ketoglutarate. Again, the enzyme is isocitrate dehydrogenase. Here, the oxidative decarboxylation reaction takes place and carbon dioxide release. Now, alpha ketoglutarate is converted into succinyl coenzymes, and this reaction is catalyzed by alpha ketoglutarate dehydrogenase complex. Reaction is again oxidative decarboxylation. Succinyl-CoA subsequently convert into the succinate, catalyzed by succinyl coenzyme synthetase, and here GDP is converted into GTP. Again, coenzyme such release. This succinate is now by the dehydrogenation reaction by the help of enzyme succinate dehydrogenase convert into fumarate, and fumarate is by the help of in presence of enzyme fumarase it converted into malate. Water molecules is added, reaction is hydration, and malate is again converted into oxaloacetate by the reaction is dehydrogenation reaction and catalyzed by malate dehydrogenase. So, except reaction 1, 3 and 4, all reactions of citric acid cycle are reversible reactions, and it was explained by Hans Krebs. The regulation of tricarboxylic acid cycle is mainly determined by product inhibition and substrate availability. If the cycle is permitted to run unchecked, large amount of metabolic energy could be wasted in overproduction of reduced coenzyme such as NADH and adenosine triphosphate. The major eventual substrate of the cycle is adenosine diphosphate, which gets converted into adenosine triphosphate, and a reduced amount of adenosine diphosphate cause accumulation of precursor nicotinamide adenine dihydrogen, which in turn can inhibit a number of enzymes. NADH, a product of all dehydrogenases in the TCA cycle, with the exception of succinate dehydrogenase, inhibit pyruvate dehydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and also citrate synthase. Acetyl coenzyme A inhibit pyruvate dehydrogenase, while succinyl coenzyme A inhibit alpha ketoglutarate dehydrogenase and citrate synthase. Citrate is used for feedback inhibition, as it inhibits phosphofructokinase, an enzyme involved in glycolysis that catalyzes formation of fructose 1,6-bisphosphate, a precursor of pyruvate. This prevents a constant high rate of flux when there is an accumulation of citrate and a decrease in substrate for the enzyme. Calcium is used as a regulator. Mitochondrial matrix calcium level can reach the tens of micromolar level during cellular activation. It activates pyruvate dehydrogenase phosphate, which in turn activates the pyruvate dehydrogenase complex. Calcium also activates isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. This increases the reaction rate of many of the steps in cycle and therefore increases flux throughout the pathways. So, student, as you all know that several metabolites in the citric acid cycle are also substrate in biosynthetic pathways. For example, those for heme or various amino acids, and through these pathways are drained from the cycle. Similarly, when the workload of a cell and its ATP demand increase, the TCA cycle must then process acetyl coenzyme A at an accelerated rate, which require an increase in the pool of tricarboxylic acid cycle intermediates. The citric acid cycle is not only utilized for complete oxidation but also for the biosynthesis of fatty acids, cholesterol, and ketone bodies. Therefore, the activity of citric acid cycle must be balanced with those of various synthetic pathways. This regulation is mainly exercised by NADH, the major direct product of TCA, and by ATP, the ultimate product of complete substrate level oxidation via TCA and the respiratory chain. Figure shows the rate controlling enzymes in the TCA cycle. It is known that key enzymes in metabolic pathways are regulated by allosteric regulators and by covalent modification for guarantee production of intermediates and products at the rate that required to keep the cell in a stable steady state and wasteful overproduction of intermediate can be avoided. The flow of carbon atom from pyruvate into and through the citric acid cycle is further tight regulation at two levels. First is the conversion of pyruvate into acetyl coenzyme A, the starting material for the cycle, the pyruvate dehydrogenase complex reaction, and the second is the entry of acetyl coenzyme into the TCA cycle by the enzyme citrate synthase. Because pyruvate is not the sole source of acetyl coenzyme A, the availability of intermediate from these other pathways is also important in the regulation of pyruvate oxidation and the citric acid cycle. The isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase reaction also regulate the citric acid cycle. Figure shows the production of acetyl coenzyme A by pyruvate dehydrogenase complex, which is required under high regulation. In vertebrate, the pyruvate dehydrogenase complex is regulated both allosterically and by covalent modification. The complex is strongly inhibited by adenosine triphosphate as well as by acetyl coenzyme A and NADH, the product of the reaction as shown in the figure. The allosteric inhibition of pyruvate oxidation is greatly enhanced when long chain fatty acids are available. When too little acetate flow into the citric acid cycle, adenosine monophosphate coenzyme A and NAD+, all of which accumulate and allosterically activated the pyruvate dehydrogenase complex. In case of plants, the pyruvate dehydrogenase complex is found in the mitochondrial matrix and within plastid is strongly inhibited by NADH, which may be its primary regulator. It is also evident that reversible phosphorylation leads to the inactivation of plant mitochondrial enzyme. The pyruvate dehydrogenase complex of Escherichia coli is under allosteric regulation similar to that of vertebrate enzymes, but the regulation by phosphorylation apparently does not occur with the bacterial enzyme. There are three strongly exergonic steps in the citric acid cycle, which are catalyzed by citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase, which are regulated by different mechanisms and each can become the rate limiting step under certain circumstances. The availability of the substrates for a citrate synthase, that is acetyl coenzyme A and oxaloacetate, varies with the metabolic situation and sometimes limit the rate of citrate formation. NADH, a product of oxidation of isocitrate and alpha-ketoglutarate, accumulates under some conditions, and when the NADH to NADH plus ratio becomes large, both dehydrogenase reactions are severely inhibited by mass action. If you see in the figure that availability of pyruvate promote the production of acetyl coenzymes, here the reaction shown in a red rectangular is the limiting reaction which control the entry of acetyl CoA to citric acid cycle. The malate dehydrogenase reaction is essentially at equilibrium in the cell, and when NADH to NAD plus ratio is large, the concentration of oxaloacetate is low, slowing the first step in the cycle. Product accumulation inhibit all three of the limiting steps of the cycle. Succinyl coenzyme A inhibit alpha-ketoglutarate dehydrogenase and also citrate synthase. Citrate block citrate synthase and the end product adenosine triphosphate inhibit both citrate synthase and isocitrate dehydrogenase. Calcium ion, which in vertebrate muscles are the signal for contraction and associated increase demand for adenosine triphosphate, activate both isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase as well as the pyruvate dehydrogenase complex. The square in red shows the role of calcium ion for the activation of these enzymes. Under normal conditions, the rates of glycolysis and of the citric acid cycle are integrated so that only as much glucose is metabolized to pyruvate as needed to supply the citric acid cycle with its fuel the acetyl group of acetyl coenzyme A. Citric, the product of the first step of the citric acid cycle, serve as an important allosteric inhibitor of the phosphorylation of fructose 6-phosphate by phosphofructokinase 1 in the glycolytic pathway. The figure shows the glyoxylate cycle, which was elucidated by Hans Kornberg and Neil Madsen in the laboratory of Hans Krabbe. In the figure, the orange reaction arrow represents the glyoxylate cycle and the blue arrow represents the citric acid cycle. Notice that the glyoxylate cycle bypasses the 2 decarboxylation step of the citric acid cycle, and that 2 molecules of acetyl coenzyme A enter the glyoxylate cycle during each turn, but only one enters the citric acid cycle. The figure shows the reactions of a glyoxylate cycle in glyoxysomes proceed simultaneously with the mesh with those of the citric acid cycle in mitochondria as intermediates pass through the cytosol between this compartment. This figure shows regulation of isocitrate dehydrogenase activity. Aspartate carries the carbon skeleton of oxaloacetate from the citric acid cycle in mitochondria to the glyoxysome where it condenses with acetyl coenzyme A derived from fatty acid breakdown. The citrate thus formed is converted to isocitrate by aconitase, then split into glyoxylate and succinate by isocitrate lyase. The succinate returns to the mitochondrion where it re-enters the citric acid cycle and is transformed into oxaloacetate, which can again be exported via aspartate to the glyoxysome. The glyoxylate formed within the glyoxysome combine with acetyl coenzyme A to yield malate, which enter the cytosol and is oxidized by cytosolic malate dehydrogenase to oxaloacetate, the precursor of glucose via gluconeogenesis. 4 distinct pathways participate in this conversion. First is fatty acid breakdown to acetyl coenzyme A in glyoxysomes. Second, the glyoxylate cycle in glyoxysome. Third, the citric acid cycle in mitochondria. And fourth, gluconeogenesis in the cytosol. The sharing of common intermediates requires that these pathways be regulated and coordinated. Isocitrate is a crucial intermediate standing at the branch point between the glyoxylate and citric acid cycle. Isocitrate dehydrogenase is regulated by covalent modification. A specific protein kinase phosphorylates and thereby inactivates the dehydrogenase. Inactivation of isocitrate dehydrogenase shunts isocitrate to the glyoxylate cycle where it begins the synthesis route towards glucose. A phosphoprotein phosphatase removes the phosphate group from isocitrate dehydrogenase, reactivating the enzymes and sending more isocitrate through the energy yielding citric acid cycle. The regulatory protein kinase and the phosphoprotein phosphatase are a separate enzymatic activities, but both reside in the same polypeptide. The intermediate of the glycolytic and citric acid cycle that lead to activation of isocitrate dehydrogenase are allosteric inhibitors of isocitrate lyase. When energy yielding metabolism is sufficiently fast to keep the concentration of intermediates of glycolysis and the citric acid cycle low, isocitrate dehydrogenase is inactivated. The inhibition of isocitrate lyase is relieved and isocitrate flow into the glyoxylate pathways where it is used in the biosynthesis of carbohydrates, amino acids and other cellular components. So friends, we discuss several reactions which are regulated by various ways in citric acid cycle. In conclusion, we can say that by looking at the changes in free energy of the reactions of citric acid cycle, it is clear that there are three irreversible steps. These three reactions of the citric acid cycle, citrate synthetase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase operate with large negative free energy changes under the concentration of product, and because the citric acid cycle is linked to oxygen consumption to generate NAD+, the citric acid cycle is regulated primarily by product feedback inhibition. Glycolysis and glycogen metabolism are under complex system of allosteric and hormonal control. Thus, the citric acid cycle must be carefully regulated by the cell. If the citric acid cycle permitted to run unchecked, large amount of metabolic energy would be wasted in the overproduction of reduced coenzymes and ATP. Conversely, if the citric acid cycle ran too slowly, ATP would not be generated fast enough to sustain the cell. This is all about the regulation of citric acid cycle. Thank you.